Thank you for joining us today on this webinar. This is part one of our webinar series featuring our microtag cell target engagement platform. Today, I will specifically talk about real-time cell target engagement, and I will make emphasis on how to interrogate challenging drug targets, how to measure cellular binding potencies, and how to integrate this with real-time machines such as QuantStudio real-time systems. Now, what are challenging drug targets? In comparison to the classical textbook examples of druggable targets such as receptors and enzymes, now the druggable or undruggable or challenging drug targets, they come with a set of challenges. We know that these targets, they lack well-defined pockets, which is necessary for classical pharmacology. They exist in complex protein-protein or protein-DNA interactions. They are highly conserved in terms of their active sites, if they do exist. And then finally, they lack three-dimensional structure outside of the cell, altogether making classical pharmacology a lot more difficult. Now, it's also important to acknowledge that challenging drug targets, they exist in the cell under various structural and functional states. Now, these structural states can be high energy or low energy substates, but we know that the function of these targets that can dictate and promote various conformational variants. As a result, these targets, they exist in the cell in a population of various substructures. And this is important for their function, and it's also more important from a therapeutic intervention point of view. Now, the question is for a drug discovery purpose, how do you engage with the relevant structural and functional states of these targets inside the cell? Now, the challenge is clear. These type of targets, they do not comply with the conventional biophysics. They require new cellular approaches, and yet we want to identify novel drug molecules, novel drug modalities, small molecule, medium-sized molecule modalities to interfere with their active sites or their allosteric sites or with their protein-protein interaction interfaces so that to interfere with them in a pharmacological way. Now, cell target engagement has emerged recently to exactly address this question. It's a cellular biophysical approach to interrogate targets within their native environment. And there are great technologies, microtag, cell target engagement technology is one of them. And this allows you to really, inside the native environment, to tease the engagement of a target with a ligand or therapeutic molecule in real time or in other type of modalities, ways. Now, the principles of cell target engagement has been established a decade ago. Imagine a cell carries thousands of proteins, and now if you expose the target or the cell for that matter, or cellular material to a temperature gradient, what happens is your perfectly structured and soluble target protein will undergo denaturation aggregation events in temperature dependent manner. Somewhere in the middle of that transition, you have TAG50, where your target exists in 50% soluble, 50% aggregated states. If you further push the temperature to a higher point, your protein will completely be aggregated and be out of solution. Now, during this transition process, your target will undergo various type of transition points, TAG25, TAG50, TAG75, and a lot more points in between, potentially mimicking many of those structural states that the target may assume inside the cell. And it's important to really design cell target engagement experiments to capture these various structural events in transitions. So cell target engagement is then again very important because it captures the physiological state of the cell and the protein. It relies on the conventional biophysical principles of protein melting, may require minimal or no modification of the target protein, can be performed with intact cells or cell lysates, and it can be used to extract kinetic measurements or thermodynamic constants. Microtag cell target engagement essentially is an enzyme complementation technology, a novel fluorescence-based split RNase technology. Now, the first thing is to identify your target. Once you know the target, you would go ahead and clonally attach a 15 amino acid tag, the microtag, to the N or the C terminus of your target protein. Once now this gives you a reporter cell for that specific target, you would go ahead and treat the cells with that ligand in whatever format necessary for your project. Then you would apply the temperature challenge in a way that to destabilize the target inside the cell to a point at which the effect of your ligand would be visible. And if the ligand is available and potent enough to engage with your target in the cell, it would rescue the target from that denaturation, and you would see a biophysical delay in destabilization of your target protein. To detect a signal, you would add the tag binding protein, which is a large subunit to the microtag, as well as a Fred substrate, which is a very specific substrate. The tag binding protein complements itself with the microtag, makes an active RNase enzyme. It cleaves the Fred substrate, releases fluorescence signal. Now, the level of fluorescence signal is directly proportional to the level of stabilization of your target protein imparted by the ligand that you're measuring. You can go ahead and use the traditional readily available plate readers to measure the end point of that signal, or you can use real-time systems like real-time QuantStudio systems to measure that signal in real time, not only just the end point, but every point in between to gain that deeper mechanistic understanding. Now, the microtag system has its numerous advantages. Its elements are heat resistant. You could use intact cells or cellular lysates, and there is no need to label your compound, or there's no need for a competitor compound. A few more words about the elements of the microtag system, the anatomy of the system, if you will. Now, on the left side, you have your target protein. You would clonally attach that 15 amino acid tag, put that back into the cell. Now you have a reporter cell. You would then detect the signal by adding the tag binding protein and the Fred substrate. By a virtue of enzyme complementation, you will generate an active RNase enzyme, which would cleave the substrate, the Fred substrate, in a way that is directly proportional to the level of stabilized and soluble target protein. As a result, you have fluorescence signal that can be detected in various type of machines. The RNA Fred substrate is a short stretch of RNA, and you can place a FAM and TAMRA donor quencher pairs to detect the signal in classical setting, or you can prefer other type of donors and quenchers to prefer various colors and various detection wavelengths. And the Fred substrate is resistant to cleavage by endogenous RNases. Let's compare a few points of traditional, conventional cell target engagement with the real-time cell target engagement. In terms of detection, now the traditional CTE will give you the endpoint after the target engagement experiment has been completed. Now the real-time CTE will give you real-time signal with every data point in between leading to that endpoint. And this gives you a lot more depth and a lot more mechanistic understanding. In terms of quantification of signal, now the traditional CTE is based on temperature of aggregation of the target protein, and then you would need to test your ligand molecules at that specific TAG, potentially leading to a bias. In terms of real-time CTE, there is no need to predetermine TAG. There's no reliance on TAG. because you would go ahead and do temperature series experiment. You would scan every temperature from any low to any high point, eventually leading a complete and biased effect of the cell target engagement that you're measuring. Engagement type, traditional CTE would rely on measuring permanent, more stabilizing visible effects, whereas the real-time CTE would give you both permanent and transient events, which would be pharmacologically important. In terms of speed of detection, the traditional CTE would is rather slow because you have to wait for that endpoint maturation. In the case of real-time CTE, you can extract the data in real time and you can see it on the screen as it happens in real setting. In terms of target behavior, the traditional CTE is built to measure the stabilization events mainly. In terms of real-time CTE, you can measure both stabilization and target destabilization events. And finally, for programmability, the traditional CTE is rather rigid. You have to go through certain procedural steps to really extract that signal. In terms of real-time CTE, that is procedurally adaptable and flexible, dependent on the stage of your drug discovery project, and also dependent on the target, the target family that you're working with. Now, we have been working and utilizing our MicroTag real-time engagement system with various real-time machines. For that matter, you can go into a variety of systems that may exist in your lab. We have been using extensively Thermo Fisher's QuantStudio machine and Bio-Rad's CFX96 well system. And we prefer these systems for their ability to be adjustable for fluorescence detection. You can use various wavelengths. These systems are highly programmable and adaptable, and they're also very convenient in terms of controllability of the heat gradient, which is a great asset for our system. And we also have recently completed an application note with Thermo Fisher Scientific using our real-time system in their QuantStudio machines. And this application note is available on the web, on our website, as well as through direct contact with our team members. How does the MicroTag real-time thermal series work? Now, on the left side, you have already established a MicroTag reporter cell for your specific target. This could be a transient or a stable system. You would put that into 96 or 384 well format. You would then add detection reagents. You would add your ligands in whatever format, in dose dependent, and then you would program the real-time system to go through multiple steps. You can program the system to be as short as 10 minutes or as long as 3 hours if you want more mechanistic and data depth to it. Once the data is completed, you would get a staggered data curvature, just like here on the right-hand side. The DMSO, the red line, essentially indicates the baseline signal of your system. And as you see here, we have started from the lowest point, 40 degrees Celsius, and we go to a highest point, 70 degrees. And as you ramp the temperature from the lowest point, you see that you get a lot more fluorescence because your target is very much stable, alive in the cell. And as you increase that temperature, your target will generate less and less fluorescence and eventually plateau. The test compound, which is a positive control for your target, would engage it with the target, stabilize it, and allow the target to be stable for a longer time at higher temperatures. Initially, you will get a lot more fluorescence. It will keep climbing up, and it will essentially at some point will slow down and it will plateau, but it will generate a lot more fluorescence compared to your baseline at certain temperature points. And this area between the curve, area under the curve, is the magnitude of stabilization of your target by the ligand you're testing. And you can repeat this for every dose of that ligand to really extract the EC50 or the engagement potency of your ligand. This system here is highly scalable, adaptable. There is no need for temperature of aggregation whatsoever. Highly automatable and programmable. You can measure target stabilization, destabilization, or abundance. And you can use the system for membrane proteins, transcription factors, enzymes, and you can flex and design various versions of your experimental workflow. Let's have a closer look on how we program these machines for detection of real-time CTE. Now, we go through multiple steps of ramp, hold, detect. During the ramp phase, essentially, we ramp the temperature at a certain speed. We arrive at a certain desired temperature. In this case, we started with 40 degrees Celsius. We hold at that temperature for a certain amount of time so that the temperature imparts its effect on the target. Then we go back to that original low temperature, 20 degrees Celsius in this case, to detect fluorescence. And then we go to the higher point. Now we have 50 degrees Celsius, ramp to 50, hold at 50, then go back to 20 degrees Celsius, detect fluorescence. And now repeat that for 55, for 60, 65, and 70 degrees Celsius to a point where you want to stop. And eventually you will stop the process, extract the data, and you have a new type of target engagement data for your drug discovery purposes. The large subunit of this real-time cell target engagement system is highly thermally resistant, and we have done the test. When you increase temperature from 25 to 60, 65 degrees, you only have a loss of around 10% in its enzymatic activity. It means that this is as good as a Taq polymerase, if you will. And it means that in the real-time microtaq system, the only element that the reagent or the element that responds to temperature should be your target protein with the microtaq on it. The rest of the system is thermally resistant and should not be heat sensitive. And then again, on the right side, you will see the staggered typical curvature you would extract from our system. You would have a baseline DMSO. This is essentially shows how the target responds to heat at certain temperature intervals, and you would see the flexing point and a point where your target starts plateauing. The test compound would essentially show how it behaves in relation to that baseline control. You would see where exactly it starts deviating from the baseline. In this case, that's 55, perhaps 53 degrees Celsius, when it starts to plateau and when the signal stops generating. And then you have area under the curve for your potency of engagement measurements. Now you can do a lot with the system, and the typical data that you get out of the system essentially could be used to extract the slopes of engagement. You can use that to extract and calculate delta Tm or delta Tag, as well as you can use that for extraction of cellular binding potencies. Now, on the left side, you have slopes extracted for every temperature for the DMSO control and for the test compound. As you can see, for DMSO, you have high signal early on, then it starts going down and eventually plateaus. The test compound starts steep, continuous steep for some time, and eventually it will plateau. You can use this slope data for generating delta Tm or delta Tag, in this case with and without the compound engagement. And then finally, if you want to measure the potency of engagement, you can calculate area under the curve for every dose of that ligand, compare that to the DMSO control, and then transform that data and plot it against the dose in whatever format, and you have the EC50. We will be talking some more about this in our future webinars. Now, the microtag system is designed to be an end-to-end platform in early drug discovery. We can go from hit discovery to hit validation. And during the hit discovery, we can essentially use it to screen through thousands of plated conventional libraries, or we can actually use this, combine the system with the DNA encoded libraries to do cellular DEL discovery. We will be talking more about this in our future webinars. And this discovers for you de novo, completely new candidates for your target of interest. After the fact of discovery, we can do heat validation. We can bring panels of compounds to the lab and we can screen them one-on-one against the target in whatever dose-dependent or format to detect for potency and selectivity. You can identify your promising drug candidates, the blue boxes. You can also identify the non-promising or low potency chemotypes, and this information will guide and inform your SAR efforts. You can go ahead and do real-time cell target engagement to understand the full mechanistic depth of that engagement in the cell across multiple temperatures. And you can also add optionally live cell imaging to gain more mechanistic insight into how the target degrades or stabilizes or redistributes itself inside the cell. Particularly important for degraders or molecular glues and novel modalities. And then finally, you can do heat expansion and optimization. So you can now generate a lot more analogs around the chemotype of your best molecules. And then you can iteratively go through multiple cycles and you can identify your novel optimized ligands. And then essentially you have a lot more speed and efficiency to your drug discovery and drug development process. The landscape of cell target engagement technologies have been evolving in the past few years. Now we have great technologies like CETSA, like Hybrid, and microtag systems. Now, it's important to acknowledge that the tag-free system the CETSA is the only tax-free system in the cell target engagement realm. And it's a great way to really measure the endogenous target, and there are great benefits to it. Um, CETSA system does not rely on a substrate because there is no enzyme complementation. It's direct use of immunoblotting directly through antibodies, whereas the hybrid and microtag system depend on a substrate to generate that signal. It's highly sensitive. Um, and then again, hybrid and microtag systems are very scalable because these are enzyme complementation technologies. Once you generate that reporter cell for your target, you can design any kind of high throughput, a very sensitive, adaptable systems. Microtag system is the only system that does not require you to predetermine TAG. There's no reliance on the temperature aggregation because our real-time application enables you to do thermal series and scan every temperature in between and determine the drug target engagement in an unbiased fashion. We can do real-time thermal series, as already mentioned, and we can work with various challenging drug targets like membrane proteins, like transcription factors, regulatory proteins, less so in the hybrid and CETSA systems. In terms of live cell imaging, we have that established. That is a topic for a webinar in the future. And this can complement your mechanistic studies. In the case of hybrid, that is less so. In the case of in CETSA, I believe that is not possible. Let's compare the two enzyme complementation systems, the microtag split RNAs and the hybrid split luciferase systems. Now, in the case of microtag, we're measuring fluorescence signal. In the case of hybrid, that is luminescence. Tag sizes are different, 15 amino acids versus 11 amino acids. Tag sequences are different. Theoretical PI for microtag is 7.7. For hybrid, that is 11.6. Aliphaticity index 20 versus 97.2. This means that in the case of microtag, the tag is thermally not stable. It means that it does not contribute to the stability of the target protein it's attached to. In the case of hybrid, it's highly thermally stable, potentially contributing thermally to the stability of your target protein. Hydropaticity index. Hydropaticity index in case of microtag is minus 1.39. It's a hydrophilic tag. In the case of hybrid, that is 0.009. That is a hydrophobic tag. Net charge is 0 for microtag, and it's +3 for hybrid. And signal resolution, we enable real-time measurement. Hybrid system allows endpoint measurement. So these are the main differences, but I'm sure there are great benefits to both enzyme complementation strategies. In the past few years, we have worked with and validated a number of various target types from various target families. We have worked with enzymes like GALC, like GBA1, KRAS, MAP kinase, PLK1, you name it. And we have optimized the system to work with enzymatic and substrate-based targets. We have tested and interrogate various nuclear or transcription factors like IRF5, androgen receptor, BCL6, FOXP3, and so many more. We have also worked with membrane proteins, GPCRs like beta-2AR, GPR75, EGFR, and a number of other membrane-bound proteins, and also optimized the system to work with various target families and to make the system sensitive and scalable, dependent on the context of stage of discovery and the target family of interest. Now, in summary, I want to make a few points about where real-time cell target engagement can be used in the realm of drug discovery, drug development. You can certainly use that for target identification and validation. You can interrogate the targets for their response to cell target engagement altogether, and you can detect their tractability, you can score them, and this would be a great start to your drug discovery programs. You can do compound screening. Now you can go into hundreds, thousands, and even higher numbers because now you have a very scalable, responsive system, and you can use our real-time CTE for large or small or medium-sized drug screening campaigns. You can validate your best compounds one-on-one against the target to determine potency, selectivity, and this would guide and inform your SAR. You can further optimize your lead compounds, and then you can do lead optimization, lead development, and this would essentially identify your best clinical candidates. And finally, you could in the future use the microtag system for during clinical trials. You could detect the level of cell target engagement from clinical samples to really inform the therapeutic and the clinical benefit of your drug discovery programs. Well, that's all. Thank you for joining us today. In the future, we will be talking more about other various applications of cell target engagement from microtag platform, and we will be back to you